So hello dear students, I hope you guys are doing well at home, relaxing and studying. So here I am talking about the variants or the types of PCR. So I'll be talking about all the different types of PCR one by one. So now let us first start with the multiplex PCR. So as the term suggests, multiplexing means more than one set of primers in a single PCR reaction. So in this one, what happens is you'll have more than one or two sets of primers amplifying the different regions of the template DNA. But you have to make sure that all the primers have more or less the same TMs and they work more or less at the same annealing temperature. So they should have annealing temperature working. They should have the sequence which have more or less the same TM, which enables them to amplify at the same uh, annealing temperature and then the extension temperature. The other important thing is it should give a different band size. So if you are going to run the PCR product on the gel, how will you identify? You should get the different size of the bands. So in this picture, it shows that all the bands are of same size. I mean, you can see that DNA is of same size, but this is not the case in the usual situation. So you try to make different size of the amplicons, and then it is better for you to identify. So it has got several applications like pathogen identification, high throughput, small single nucleotide polymorphism genotyping, then mutation analysis, gene deletion analysis, linkage analysis, DNA, RNA detection, and forensic studies. So now next is the VNTR PCR, which is the variable number of tandem repeats. Variable number of tandem repeats PCR. So VNTR is something which actually is present within the genome. So different genes might have the VNTR polymorphism. That means you might have a stretch of tandem repeats which are present in n number of repeats. It could be 2 times, 3 times, 4 times, 5 times repeat. So this particular picture shows, this is a gel picture of IL-1 receptor antagonist polymorphism. So in this one, different alleles have different number of particular repeat. So allele 1 has 4 repeats, allele 2 has 2, allele 3 has 5, allele 4 has 3, and so on. And a particular individual can have only just 2 alleles since the individual is a diploid. So this is this is one of the individual's genetic polymorphism of VNTR of IL-1 receptor antibody. So this is 1, 1. This is 1 and 2. This is 1 and 3. This is the molecular weight marker. This is 1 and 4. This is 2 and 2. This is 2 and 4. So this is very important in the genetics and the basic research as well as forensic studies like DNA fingerprinting. Then comes the asymmetric PCR. So asymmetric PCR is something where you use the unequal proportion of the two primers. One of the primer is sorry for the disturbance. So let us continue. So one of the primer is enriched, that means it is abundant, whereas the other primer is in a limited number, because we preferentially want to amplify this particular fragment. So you can see the blue colored one is in the more number, whereas there is very less double stranded fragment. So this is usually done for designing the hybridization probe, which is usually single stranded. or even for the DNA sequencing where you need to amplify the single stranded and you want to know, for example, the forward strand reading sequence. So if you add both the primers, then you will end up getting the noise. That means forward and reverse both. But you want to enrich it with just one primer so that preferentially one single strand is synthesized. So the amount of primer for the targeted strand, so this is a targeted strand, is too much high as compared to the non-targeted strand. Now comes the nested PCR. So nested PCR is done in the situations where, for example, this is a target sequence which we need to amplify. And whenever we design a primer to this target sequence, it has problem in getting amplified. So this region is difficult to amplify, possibly because the primer designed to amplify this region might bind somewhere else in the genome. So what usually is done is you first design an outer set of primer which preferentially amplifies a bigger region including the target region, and then the second round of PCR you do this as a template and then amplify the shorter the main target region. So then you will be it will be easier for you to amplify this particular target sequence. So this is what is called the nested PCR. Then comes the very important one, the quantitative PCR or the qPCR. So it is used to measure the specific amount of DNA or RNA in a sample. And usually the amplification graph is something like the growth curve of the bacteria. If you want to understand. So you have a lag phase, then you have a log phase, and then you have the stationary or the plateau phase. There's no death phase, there's just stationary phase. So in the exponential phase, what happens is there is increment in the number of copies of the DNA. So usually actually the amplification or the amount of product is usually measured at the exponential or the log phase rather than the end stage or the stationary stage. And there are special thermocyclers used for the real-time PCR machine or for the qPCR. So the qPCR monitors the amplification of the target DNA during PCR in the real time. That means when the amplification is going on, they will show you the graph, online graph that it has started to amplify. So there are different methods of the quantitation of mRNA and DNA. The direct methods include the northern blotting, in situ hybridization, and also it includes the PCR amplification. So another blotting is a blot based, in situ hybridization is a tissue based, and PCR is an in vitro where either you can perform the regular reverse transcriptase PCR where you convert the mRNA, extracted mRNA from the tissue to the complementary DNA, and then you perform the PCR to quantitate the expression level. Then you have the real-time PCR for the amplification, and also you have the microarrays, which are the miniature real-time PCRs, which gives you and those are present in the chips. So it is easier to perform multiple samples in one shot. Now, why people prefer using the real-time as compared to the traditional or conventional PCRs? What happens for the agarose gel is, it is actually having low sensitivity, low resolution, and they are not automated, means you have to do everything manually. And there is just size based discrimination, as you have already run the agarose gel of the PCRs, RAPD and RFLPs. You can see you tend to see different sizes, and the results are not expressed as the numbers in the sense you just see positive or negative. It's not the quantitation, that means how many copy numbers of the DNA is present. So, and it is mostly based on the personal evaluation, so it differs from person to person. And ethidium bromide staining is not very quantitative, so you can see in this particular case and in this one, so the thickness of the bands having the 10 copy initial copy of the template DNA and having the 50 initial copy of the template DNA is more or less the same. You can see the thickness of the band is more or less the same, so you don't know what was the actual template copy in the original sample, because you tend to get the same thickness of the band. That means it does the endpoint analysis. That means when the PCR is almost over, then you stop the machine, run the agarose gel. So after the whole amplification cycle is done, that means the exponential, after exponential you get the plateau. So in that case you run the gel. So everything would be more or less of the same size. The bands you see of the same size, so which actually does not evaluate the actual copy number of the template present in the sample. So as you can see, if you use the real-time PCR, what happens? So this particular sample started amplifying earlier, so it started showing the exponential phase earlier. So it has 10 to the power -1 initial template concentration. It has got 10 to the power -2, so it started after some time. -3 started even after that, -4 after that, and -5 even after that. And this is the negative control where there is no amplification at all. And you can see this particular line. This is called the threshold cycle. So this determines the CT, threshold cycle. So when you draw a perpendicular line to that, so you can roughly see the threshold cycle for the 10 to the power -1 concentration of sample is around 22. CT is 22. Here it is CT is 24. Here it is 27. Here it is 31. And here it is 34 or something. So you can see that endpoint, most of them reach more or less the same. So the conventional PCR actually measures at this particular point and not at these points, the exponential phase. So that is why people choose using the qPCR, where you actually get to know the real-time amplification, and if it gets amplified earlier, you have a more copy number. The principle is based on the detection and quantitation of a fluorescent reporter. So instead of measuring the endpoint, we focus on the first significant increase in the amount of PCR product, and the time of increase correlates inversely to the initial amount of template DNA. So as I have told earlier, if you have more DNA, it will start amplifying earlier. So that is inversely related. And this is the thermal cycling block. And your sample will have some fluorescence, which will be detected by the CCD camera. So when the light source is given, so it will fluoresce, which will be captured by the CCD camera and represented on your screen. There are different chemistries used for the real-time PCR machine. The first one is a non-specific detection, where you use a dye, which is a fluorescent dye, which is used as a reporter. It is called the cyber green. So it is green in color. So it works more or less same as the ethidium bromide we use. So it binds the minor groove of the double-stranded DNA. So that means it can bind any of the double-stranded DNA present in the sample, be it your template, be it your amplified product, be it your primer dimer, which is a non-specific amplification. So it binds everything. So it is a non-specific detection. The second is a specific detection, where the fluorescent reporter probe is used, which are called the Tachman probe. So this is a Tachman probe based method. This is a cyber green dye which binds to the minor groove of the DNA. So in the Tachman chemistry, what happens is you use the forward and the reverse primer with the template. So, and also you use a probe which binds somewhere in the middle of the template. And this probe is bound to the reporter dye on one hand and the quencher dye on the other hand. So what happens is, when the DNA polymerase starts amplifying beyond the primer, so this polymerase has got the 5' exonuclease activity, 5' to 3'. So it has got the strand displacement activity. So what it will do is, so this has because of the exonuclease activity of the polymerase, this would be chopped off and releasing the reporter. So the idea is to release the reporter from the quencher so that there is a fluorescence in the reaction. When the two are situated close together, like the reporter along with the quencher, so this quencher, the function of the quencher is to stop the reporter dye to fluoresce. So once, because of the nucleus activity, this reporter is released in the solution in the PCR mix, then it is able to fluoresce because the quencher is no longer associated with it. So this gives out the fluorescence or the positive signal. And since it comes out from the probe which bound to the template, and this can only be removed by the DNA polymerase activity, this is a very specific chemistry. The other thing is, the TM of the primer and probe should be different. Like the primer has got a TM of around 58 to 60, and the probe should have a higher TM. That means 68 to 70 degrees Celsius, so that it can still attach when the primer is starting to extend. The other chemistry is the cyber green chemistry. In this one, normally when the double-stranded DNA is found, it just binds to the double-stranded DNA in the minor groups. So problem with cyber green is that it binds non-specifically to any of the double-stranded DNA. So it can be from the non-specific products also, which might give you incorrect quantitative results. So to sort out this thing, there is another necessary step which is done after the cyber green based PCR. You do a melt curve analysis. So melt curve analysis actually tells you whether the amplification is specific or non-specific, whether it is just the contamination or the primer dimer or the specific product. So the real-time PCR actually gives you the access to the high quality geometric phase data, and there are several applications of qPCR, which includes gene expression, that is mRNA to cDNA, and then the quantitation, and then gene copy number, how much is the copy number of that gene present, could be quantitated. Then viral load, how much is the load of the virus. Drug therapy, DNA damage. So for detecting the COVID-19 also, we use the qPCR based method. So in Indore also and all over the world, people use this method. Here first the swab sample is taken. nasal or the oral swab sample is taken, and the lysis of the virus is done, RNA extraction is done, and then RNA is converted to the cDNA, and then this cDNA is used for as a template for the amplification and detection of your viral genome, viral mRNA, which is the gene, not the mRNA, viral genome. And if you see early peak, then you see this is a positive case, and this is COVID-19 positive. And the whole procedure takes around 6 to 7 hours in standard condition. Then comes the hot start PCR. So hot start PCR is like I have already told you about hot start polymerases. So hot start PCR can be done either with the help of the hot start polymerases, or if you don't, if the lab cannot afford to buy the costly polymerases which are specialized, like the chemically modified or antibody tag, then you can still use the normal polymerases used in the PCRs. What you can do is, after the denaturation step is done, you can pause and then add the tag DNA polymerase, and then again go ahead with the extension, annealing and extension steps. But this would be a tedious job, so better you go for the chemically modified or antibody modified specialized DNA polymerases, which needs higher temperature with some longer time to get denatured, to get activated. Then comes the touchdown PCR. So touchdown PCR is something in which the annealing temperature is actually set around usually 3 to 5 degrees Celsius above the TM. So this annealing temperature is set above 3 to 5 degrees Celsius, the TM, and then it is amplified in the higher annealing temperature for initial 10 to 15 cycles, and then later and what happens is, here annealing temperature is kept, for example, plus 10 degrees Celsius, TM plus 10, and with subsequent cycle, like the first cycle it was at around 65, then the second cycle it will be around -1, that means 64. Then the next cycle it will be around 63, then 62, 61. So 10 to 15 cycles, it will reach around 50 degrees Celsius annealing. And then the last cycles of around 20 to 25 cycles will be performed at around 50 degrees Celsius. So the idea behind using this is we want very specific DNA to be amplified. So in that, if you increase the annealing temperature, only highly specific regions gets amplified, removing the removing the uh, parts which should not get amplified or which are not our target sequences. So this touchdown PCR is important in many cases. Then comes the assembly PCR. So assembly PCR is something you artificially construct a long stretch of DNA from the small oligos. So it is the artificial synthesis. So this artificial synthesis of long DNA requires the multiple oligos, which are around 50 base pair in length, and they have an overlap of around 20 bases. So what happens is, initially they are made to extend, so that for around 30 cycles, so that these processes extends. And after extension, what happens? The gaps are almost filled. The 20 base pair overlaps, and then what happens is additional 23 cycles with the end primers is done. So you have the forward primer, you have the reverse primer, and then the whole long stretch of the DNA is synthesized. Then comes a very common one, the colony PCR. So I have taught you about the transformation. In the transformation, what happens is foreign DNA is captured by the bacteria. And if you want to know whether the bacteria has got the foreign DNA or the plasmid, then what you need to do is do a PCR, extract the DNA, do a PCR, and then find out if the construct is present or not in the extracted DNA. Another simpler thing you can do is you isolate the, you take the colony and make a replica plate and use the same tip to add the colony directly into the PCR mix, and then do the PCR method. And what you can do is you can see after running the gel, you can see the band for the positive transformants, because the primers are targeted towards the construct, the positive construct only. And only thing is it is PCR, but with a little higher denaturation step, so that the bacterial colony, from the bacterial colony, the DNA is extracted out during the PCR itself. Then comes the cold PCR. So cold stands for co-amplification at L lower denaturation. So COLD denaturation temperature PCR. So this actually is used for identifying the mutant types of alleles in comparison to the wild type. So you have a mixture of, you have a sample having a mixture of both mutant and the wild type. And the mutant is lesser in number and wild type is more in number. So how will you preferentially identify the mutant one? So what you used to do is, you first go for initial, intermediate annealing at around 70 degree, where, so this is a mutant gene and this is a wild type. So when you go for intermediate annealing, so there would be some heteroduplex formation, like some of the wild type might bind to the mutant allele, mutant gene. Then this, since there is a mismatch, so this property utilizes that the denaturation of the heteroduplex having a mismatch is at a lower temperature as compared to the perfectly matched one. So if a perfectly matched one denatures at 94 or 95, this will denature earlier, around 86.5 or 86 degree. So at 86 degree, the denaturation is done, and then primer annealing will be only done to the mutant one, mutant in the sense the heteroduplex, and you can end up getting the preferentially mutant allele amplification along with some wild types. But this is the step to, since you allow the heteroduplex to form, so this actually is a benefit. When you denature at lower, so only heteroduplex will separate out, and the primers will only bound to the separated out strands and preferentially added or amplify the mutant alleles. So this is used for the detection of mutants in the cancer from tissues and body fluids. Also monitoring the outcome of the therapies and cancer remission or relapse. So this was the protocol which I have told you. Then comes the ligation mediated PCR. So ligation mediated PCR is the one where you use the linkers at the ends of the DNA fragments, of which you don't know the sequence. So for an unknown DNA sequence, what you do is you digest it to make the sticky end. Then you add the linkers, and since you know the linkers sequence, you can have the primer specific to the linkers, and then it is easier to identify. So this example shown here is you have a, viral DNA integrated into the host genome, and you want to know which part of the host genome it is integrated to. So you partially digest to the whole DNA into the sticky ends. Then you add the linker and do the ligation, and then you design the primers to one towards the uh, viral genome end and one towards the linker, so that you get to know where exactly in the human genome the viral DNA has integrated. So this is used both for also for the DNA sequencing, genome walking, and DNA footprinting. So DNA sequencing, I'll be telling you in my subsequent lectures. Genome walking, DNA footprinting, you know DNA and protein binding. The transcription factors I have told you, DNA is one footprinting, so that is the same. And then comes the genome walking. So I'll tell you what is genome walking. This is a genomic DNA, and you just know a small stretch of that genomic DNA. So this is the known sequence. So in genome walking, what is done is you digest restriction by the restriction enzyme, and use the adapters to ligate the ends of all the fragments. And after that, you fill the ends with the polymerase. And then what you do is you design the PCR primer, one specific for the linker, the other specific for the null sequence. So then after the amplification, you can go for cloning and sequencing analysis, and you get to know this particular sequence. So initially you knew only this much sequence, now you get to know this much. Then subsequently what you can do is again restriction digestion, and then again perform this step so that you go back and get the further sequence to that. So upstream sequence of this. So this is how the genome walking is done. Then comes the methylation specific PCR. So methylation specific PCR is, the human DNA is usually methylated as the CPG islands. And what usually is done is, so since this CPG islands are methylated, so this particular feature is used. So what happens is the target DNA is first treated with a sodium bisulfide, which will convert the unmethylated C bases to U. So unmethylated C, it can convert to U, but the methylated one, it cannot. So then it converts to U, and then during the PCR, it will bind to A in the complementary strand. So the two amplifications are carried out on the bisulfide treated DNA. One is a set of primers that anneal to DNA with C, which corresponds to the methylated cytosine. And remember, methylated cytosine cannot bind the get converted to uracil, but the unmethylated can. So another set will anneal to the DNA with U, which corresponds to the unmethylated cytosine. So within the reaction, what will happen is, if you have unmethylated sequence, so you will have C, which will be converted to U during bisulfite, and then methylation specific PCR will, you will see that it will complement to APTT. Whereas in the methylated sequence, the C will remain as C even after the bisulfite conversion, and you can find that it is CCC. Then comes the, I guess, the last one, which is inverse PCR. In this one, what happens is you have a long stretch of DNA, and just the middle of this stretch is known, whereas the others are unknown. So what you do is you will digest the ends with the light digestion with the restriction enzyme, so that only the ends digest, not the in between. You'll end up getting many fragments along with this one. What you will do is allow it to self-ligate. So once it self-ligate, it takes a loop form. Now, since you already know the sequence, then you use a digestion restriction enzyme and to cut open it. So now what you can do is you can see the known sequence towards the end. And since you already know the ends, you can design the primers and amplify this particular region and get it sequenced. So easy, right? So this is all about the PCRs. So I have given the references of lot many materials you can refer to. Get back to me for lot many queries I guess you'll be having. Thank you so much. God bless you.